Hi, everybody. Thank you for joining us today. We are very happy to be hosting Dr. Woolf. He was born and educated in South Africa. However, he's currently the F.M. Kirby Neurobiology Center Director at the Boston Children's Hospital and is also a neurology and neurobiology professor at Harvard Medical School, along with being the co-founder of a couple different companies. He works on adaptive and maladaptive plasticity of the nervous system. Thank you, Dr. Woolf, for joining us, and I will turn it over to you. Dr. Woolf has chosen to do a discussion format today, so all of our participants should have the ability to unmute themselves if they choose, or you can continue to use the chat function. If you raise your hand, I'll be able to call on you and kind of help moderate discussion, and Rob is going to be monitoring the chat for any questions. So you can email him or chat him directly or send it to the group. All right. Go ahead, Dr. Woolf. Thank you.
Great. Perhaps if people don't mind, they can switch on their video cameras, because otherwise I'm just speaking into names on black images. Yeah. It's always nice to know there are real people somewhere. Great. Well, as I understand it, this is an opportunity to discuss career directions for you as you contemplate your future. And certainly the perspective, maybe just to begin with, is that when I went into academic biomedical science, I'm an MD PhD, so I did both, but having got my medical degree focused on neurobiological research. And certainly the first sets of students, graduate students and research fellows that I mentored, there was almost zero expectation of people going into industry at that time. There was a clear separation. Those people who went into industry started off in industry and stayed there, and those people who did academic research tend to stay there. It wasn't completely hard, but that was the general rule. And things have changed enormously in the last decade in particular, and obviously, as I'm sure you're aware, that Boston and Cambridge have now become one of the big biotech hubs, and in consequence, we find that many of our grad students coming in to Harvard Medical School are very interested in careers in the biotech, and many of our research assistants are poached from us by large biotech companies. So we realize a big part of what we're doing is training people to help the biotech system, but it's also it's a two-way relationship. It's on the one hand, they're using our academic skills, our innovation, our approaches, but they have the focus on translation and the development of new therapeutics, new biomarkers, new devices. And there's a kind of rigor there, but also complicated by the fact that almost always in the biotech world, there are investors who are looking for return on their investment. So the academic world and the biotech world are quite different, but increasingly there is overlap. And for me personally, I run a lab, and of my recent graduate students, let's say about of the 4 or 5 of my most recent graduate students, 2 have gone into industry, and 1 is looking for a job in industry, and 2 have gone into academia. So it's a whole new world, and that means as a mentor, part of what you need to do when you train people is to give them those skills and that exposure for them to make a choice, whether how they see their career, and if they are interested in industry, how to prepare for it, and what are the skills they'll need, what are their expectations, and also how to introduce the highest quality science into the biotech world to make sure that they are at the cutting edge. And then, of course, there's the issue that hopefully when you do academic work, there are chances you may discover novel intellectual property that potentially could be licensed by biotechs, or you may be getting involved in actually starting up biotechs. And that too is a whole new skill that I'd say in the first half of my career, I didn't file a single patent and had no sponsored research from any pharmaceutical companies. And it's been very different in the second half of my career. And so I'm here as a resource to explore with you these new worlds, the intersection between the academic and biotech world, and I'm happy to either give you examples of this or to answer questions. And I'd prefer this to be a discussion rather than just me giving you my viewpoint. So why don't we start by people telling me about what your plans for the future are, at least as you see it at the moment. And then, of course, I expect the normal Zoom silence when you ask that kind of question. Who wants to start?
I can start us off since I was able to have the privilege of inviting you. So I am just a second year grad student. So I'm still very much open to exploring different options while still understanding that I'm probably not going to end up in academia, which is why I'm part of this group. And I think there's a whole world out there that even still in current graduate school that is not really supported, or there's a lot of information. So the point of this group was to kind of explore career options. And I've done that not only by attending events, but also by seeking out different speakers to come and give presentations to the group. So I currently study Prader-Willi syndrome using stem cell and stem cell induced neuron models. And I plan on probably not doing that after I leave graduate school, but I'm definitely enjoying it while it lasts.
Great. Well, how about, since you were brave enough to get us going, the kind of thing that I think we should be thinking about, given your the nature of your research project and your thesis work, is that we need to look critically at drug development. The standard model for the last 3 decades has been identify a target that is involved in the disease process, identify it by virtue of the genetic mutations that reveal the disease, or by experiments where you manipulate the gene by overexpressing or knocking it out, or mutating it, and you get some disease phenotype. And then you isolate the target and you screen for compounds that interact exclusively with the target. And then you test for efficacy in preclinical models, and you use medchem to make drug-like molecules that interact with the target exclusively, but are still orally bioavailable and are stable and have the appropriate PK and safety, etc. And then you enter into the IND enabling studies. At that stage, the investment goes up, so you need either a partner in industry or a biotech that will take it. And you put it into safety studies in humans. Hopefully it's all safe, and then you go into humans looking for the first sign of efficacy. And this is where the model begins to fall apart, because costs, by the time you get there, it's now many millions of expenditure. And the expectation is that your target is a key player in the disease process, whatever you're interested in, and that the drug, which is target exclusive, by virtue of acting on that target, is going to produce some kind of efficacy signal that will either prevent the development of the disease or stop its progression or produce repair or whatever it is that you think may be relevant. And the reality is that the vast majority of phase 2 trials, efficacy trials, fail. There's just no efficacy signal. And there are many reasons for this. Maybe there's inadequate target engagement, maybe the wrong patients have been selected, maybe the wrong outcome measures. There are many reasons that they could fail. But I think one possibility is that the drug development model is not the right model, and that if, for example, in the case of your thesis work, instead of going for a single target reflecting the disease, we embrace the fact that diseases may involve multiple targets, may involve complex interacting targets that may involve multiple signaling pathways, and that rather than going for a single target and putting all your money, all your eggs in a basket, maybe it would be better to screen against a disease model, ideally a patient-derived neuronal model, for example, a phenotypic screen, which captures, if it captures the disease, then there's a real chance that maybe you'll find a compound that will act to normalize the disease and turn the disease cell to a healthy cell. And it may do so by acting on multiple targets, which is the industry says, oh, no, we can't deal with that. Because when we do medchem, what we've got to do is to optimize the hit into the drug-like molecule. We've got to make it more potent and more selective. Whereas when you do a phenotypic screen, you don't necessarily know, and often don't know, which target your compound is interacting with. And basically, I think this is where modern technology, for example, neural networks and machine learning and AI, are enabling us to overcome that problem by saying, well, even if we don't know the target, if we know the action of the drug and we can have good, reliable measures of efficacy and adverse effects, then we can eliminate the target-based model and come up with a disease-based model, which is much more complex, much more difficult. But maybe, if things work out, the phase 2 trials, instead of being at best a 1 in 10 success, might be a 50% success. And instead of our drugs being single target, beautiful drugs that act on a single target but have lack of efficacy, maybe there'll be complex polypharmacological drugs that act on multiple targets, which are much more difficult and complex. But many of the most commonly used drugs, the old-fashioned drugs, are exactly that. They have actions on multiple targets, and that partly is responsible for their adverse effect profile, but it's also partly responsible for their efficacy profile. So maybe what we need is, and that's the point I was trying to make, is that the relationship between academia and industry is that industry is driven by the need to produce new treatments. It's driven by the need to provide shareholder value. But sometimes that means they take a path that is not so risky, that is, they think, the right and correct path, the one-way path. And sometimes you need to shake it up. And hopefully this is why we need to develop a means of interacting in a way that academic risk-taking, a different way of thinking about things, a new solution can hopefully upend the standard industry approach. So don't be too soon, Rachel. Don't give up your iPSC-derived neurons too quickly, because it may be the future.
Yeah, it's interesting that you say that, because my lab specifically, while Prader-Willi is still in much more infancy as to understanding and developing possible therapeutic approaches, its sister disorder, Angelman syndrome, is a little bit further along, and our lab does use neuron models to screen drugs for pharmaceutical companies, or I guess maybe not for, but in partnership with pharmaceutical companies. And they do the brute work of, like, we have all these different small molecules, and then they ask us, it works in a mouse, what do you think about it in human cells? So I think those partnerships are definitely forthcoming. I just think they're rare. I don't know in the academia scene how it's looked upon, but I think there should be definitely more partnerships like that working together.
Yep, absolutely. Who's going to volunteer next?
I can go second. So I'm Rob. I'm on the other end of the spectrum. I'm a fifth-year graduate student, also at UConn Health. I co-founded this group where I saw a need where students and postdocs need to learn about what other options they have. A lot of people choose to go the postdoc route because it's safe. We are aware of what the academic bubble really kind of is. And so we created this group, myself and two other postdocs, Julia and Britt, and now we have a whole team of graduate students working with us at UConn Health as well as Boston College, and we're still expanding. But my own work is in the neuroscience field. I study how the accumulation of aged cells impacts neurodegenerative diseases and normal aging. And the really fascinating thing about the brain is that it's so different from every other organ. And in terms of therapeutics, we're looking at how drugs target these cells that accumulate. They're called senescent cells. And so usually these drugs are discovered because people create a whole group of senescent cells and they take out maybe like a piece of a liver and then irradiate them. And now they're senescent and they sprinkle the drugs on and they say, alakazam, it kills senescent cells. But we're seeing that if you give these drugs to healthy animals, you have these nasty side effects. So I guess coming from the point of like an academic screening these molecules after they're already being used, I guess, for this purpose, like how do you get into, I guess, designing therapeutic companies around a basic science idea and then being able to translate that? So you co-founded two companies and you're a professor at Harvard Medical School. So I guess finding the time and how you created two companies is what I'm curious about.
Right, so the two companies are Nocion Therapeutics and QurAlis. Nocion Therapeutics is a company based on a strategy of silencing nociceptors, the pain-initiating fibers. And this is work that I did with Bruce Bean at Harvard Medical School. Basically, we went to a neuroscience dinner one night and we had valet parked our cars and there was a single person picking up the cars, and we suddenly realized it's going to take an hour or so for us to get our cars because they were right at the back. So we were just standing there waiting, and we set ourselves a task. We said, okay, local anesthetics are non-selective. They block every excitable cell, every cell in the nervous system as well as the cardiovascular system. And at least in terms of their use as local anesthetics, they block all sensory neurons, all autonomic fibers, all motor neurons. And how about, before we get our cars, let's come up with an idea to see if we can produce a local anesthetic that only blocks the pain fibers. If we could you go to the dentist and you have an injection and you'll get analgesia, but you won't be numb, you won't dribble, your face won't feel paralyzed, etc. And it was a fun game, and we played the game, and we actually came up with an idea. And it is one of those bizarre things where I went back to my lab, and there was a fresh postdoc who had just arrived from Israel, actually, and he was an electrophysiologist, hadn't done anything. I said, we've come up with this idea, Bruce Bean and I, we can actually, basically the idea was lidocaine as the prototypical local anesthetic diffuses through membranes, then blocks sodium channels from the inside of the cell and switches off the sodium current, and that's how it eliminates excitability. And our idea was how to have a compound that would only act on nociceptors. And the idea we came up with is if we took lidocaine and made it permanently charged, it couldn't diffuse through membranes, but if it was small enough, it could enter those cells that have large pore ion channels, which happen to be just the nociceptors. They have TRP channels, for example, like TRPV1, which is activated by capsaicin, the ingredient in chili pepper. And these are large pore channels. So maybe we could use these channels as a drug delivery portal to get into the cell. So in fact, there is a quaternary derivative of lidocaine called QX314, which has been a standard tool as a means of blocking sodium channels. You introduce it intracellularly, you put it into your pipette, and there's a way of just, if you want to study potassium channels, you just block all the sodium channels with QX314. So we just said, we expect if we put it outside a cell, it will be inactive, and that turned out to be absolutely true because it's permanently charged. It can't diffuse through the membrane. We then added it to dorsal ganglion neurons and exposed them to capsaicin, which activates TRPV1, and lo and behold, all of those cells were silenced, and the cells that didn't express TRPV1 were unaffected. This took about a week, and then we repeated the data. It's one of those rare occasions. We then sent off the paper to Nature about literally 2-3 weeks after we had the idea. It came back, and the editor said, I've read this paper, it's quite interesting, but I need some additional data. So they came up with data that took us about another month to do. We resubmitted it, went off to 4 reviewers, and they all came back. They loved the paper, except they said there's one section they didn't like at all, and that was the experiments that the editor had made us do. Anyway, so it's one of those totally bizarre things where we had an idea. The actual experiments were relatively simple and direct. The answer turned out to be exactly as we anticipated. This was in 2007, and it then took something like 10 years before we could reach a point at which we could get potential investors, set up a company, come up with an indication, which surprisingly, instead of the obvious one going for release of pain, turned out to be cough, because nociceptors innervate the airways and they initiate cough. And the reason why it was cough is because investors look for an area where there's a big clinical unmet need, because that's where they'll get a return on their investment. And it's for a topically applied drug. Inhalation is well known as a way of delivering it. So it just, again, totally unexpectedly, we never thought of cough initially as being the indication. And actually, the company has produced a charged sodium channel blocker, and it's finished its phase 1, and the very first patients are being recruited, and so we'll know by the end of this year if it works or not. The point is, at the time we had the idea, the playful idea, how can we target nociceptors? How can we selectively silence one cell and not another? We weren't saying, let's do this to develop a company. It was just, how can we devise a strategy to come up with a new way of developing tool compounds that can have a selective action on one cell over another. But we'll see where it goes. But and in the end, if you are going to develop something that has translational potential, then you've got to get into the complexities of protecting the intellectual property, filing patents, which then drives you down the route, which are the best sets of compounds. You've got to do screens for them. You've got to get funding for that. The NIH are less keen, so we have to go to the DOD to get funding. And there comes a point where you say, well, at what point should this be licensed off to a company so they can get startup investment to develop it, or do you develop it in-house to the point at which you actually have a candidate? And those are things you can, a big difference between giving an idea very early and giving it later when you've developed it almost to the point at which it's ready to go into IND enabling studies. And there's a big steep learning curve because we are not taught how to file patents. We're not taught how to write to get an investment presentation to venture capitalists. They have a very they are interested in science, but they're very happy for that to be summarized in 2 slides. What they want is the business plan and the how much it's going to cost and what the milestones will be and who are the competitors. You know, it's a completely different mindset. And again, we don't formally have, well, we haven't until recently had formal teaching of that, but I don't know what it's like in UConn, but Harvard Business School used to be, again, completely separate from the medical school, but now there are innovation courses teaching people what is innovation, how to prepare for work in the company or starting up a company. And actually, one of the major inputs is less the academic input from the business school and more people who've been in industry, who've experienced it, who come and give lectures.
Absolutely. We have a question in the chat regarding a point that you made on AI in pharma. So the question is, do you see AI replacing major research roles in basic biology, or do you see it more as a tool that will complement basic research?
I think it'll never replace it. It is another tool that can be used. The beauty of it is that it's unbiased. You can train up an algorithm so that if, for example, you want to design a compound that had action on one G protein coupled receptor and interacted with an enzyme, just for argument's sake, that you thought in order to interfere with the disease process, you needed both those activities. That's the kind of thing that'll be very difficult for a med chem, because their training is just to deal with docking of a compound with a unique single structure. And now with cryo-EM, it's great. You can see exactly what the structure is, and you can predict which kind of compound they fit into the active site and how to change it to optimize that fit. But if you have 2 targets that you want a compound to interact with, and you run screens and you start finding compounds that have this profile, and you have AI-based analysis, the learning, the computer learning can help you predict what changes, and they're not based on docking. They're just based on the learning experience of the algorithm, saying these compounds didn't work, this one did, and this one, and will start suggesting things that you test. So it becomes just a new way of generating new sets of scaffolds that potentially will have the profile you're interested in. So it's different from just screening for them until you find something. It's something where it's driven based on the actual information that you get. And so, you know, in previous screens, things that are not formally a hit, you just throw away. But within an AI-based analysis, you actually learn by it. This didn't work. That's actually almost as valuable information as something that did work. You keep, and the information remains there. So it's just a way of just, it's a different approach. It's not going to replace anything, but it certainly adds in a strong way. And obviously it's not exclusive to screening. There are many features of capturing many aspects of biology.
Okay, so I'll go next. I just want to follow up. So my name is Neeraj Singh, and I am at UConn Health. I'm working in the field of neurodegenerative diseases, Alzheimer's. So back when I was doing my PhD, I filed for a patent for my PhD research work, and it got approved recently, that patent-wise. Now I want to carry it forward, that research, and how should I move now that I'm a postdoc? So I don't know, that research work is not being followed by my PI right now. So how can I move that work forward, or enough to push it towards make it interesting enough for industry to, or I can contact industry to see whether it's being applicable. It's for the treatment of Alzheimer's and Parkinson's disease. So,
Yep. Okay, well, obviously an issued patent is great. I mean, it's also public knowledge. The patent is published, and so presumably people have accessed it and looked at that. I think it's important for you to get advice by people who work in the field to get a sense of the value of your IP. But I can just again tell you a story. I had a postdoc in my lab who came from the University of Minnesota, and he, like you, had filed a patent for his thesis work. And the work he did in my lab was completely different. So he did a full, but when he finished his postdoc, he started a company, and he took the patent from his PhD thesis, and that was the basis for his new code that he developed after my lab. And I had no involvement in it because it was his PhD thesis, not his postdoc research. So that is one possibility. So you've got to ask yourself, do you want to be the player here, in which case that you may have to be involved, or do you just want to license it, in which case you need to do research into which of the companies working in Alzheimer's, what are their interests, who has the interest closest to you, who can give you advice to help you on the licensing. Ideally, in an ideal world, I don't know which university you did your PhD thesis, but that tech transfer office should be the one that should be helping you with that, because they own the IP and they should be helping you in the marketing of that. To be honest, my experience is they are very good at collecting the royalties, but are less good in actually helping you, as it were, make a convincing case why your IP has value. And in the end, you have to go and make presentations and convince people that this represents something that they should be interested in. You've got to also realize that even though you have IP, that there is also freedom to operate, in the sense that a company could take your IP and actually use it and test it until they were convinced there were value. And they're not going to license it from you until they think they need that freedom to operate. And the reality is that if it works really well, they'll say, well, let's try and change your discovery in such a way that we don't need your patent. And that is an unfortunate reality.
right.
I happened to meet the head of the tech transfer office at Harvard just a couple of years back. And he was saying that over the last 50 years or something, that I don't remember the exact numbers, but for argument's sake, something like there'd been 1,000 licensing agreements with Harvard that included composition of matter. These were potential compounds that could be used for particular diseases. And if those compounds were used, then Harvard would get substantial royalties and milestone payments. And that's one of the big motivations to capture IP and put it into patents and then have licensing agreements. And he said of those 1,000 occasions where companies had bought the license, there were only 2 cases where the actual compound was used as a drug and royalties were paid, because the standard model out there is that companies try and bend the rules to avoid the IP, and that's the reality. So a new strategy in IP now is to try and capture the whole universe, so that startups now, when they got a new strategy, not only do they try and protect their lead compound that will hopefully become the clinical drug, but they try and patent the whole universe around it so that competitors can't get it. And that takes a lot of money and a lot of effort. And so it's a strange, unfortunate aspect of the universe we live in now, that it's actually relatively easy to avoid a patent. That's why if you look at successful drugs in any field, it's the large pharma, there will always be 4 or 5 people all going after the same indication with a similar target. That even though there were patents that may have appeared to be exclusive. These days, the patent office will not allow you to patent inhibition of a particular target. It's got to be a specific compound interacting with that target. But more recently, they've relaxed a little bit. And in fact, we had a patent issued that was surprisingly broad, involved activation of potassium channels for neurodegenerative diseases. And we based that on a discovery of a particular compound acting on a particular potassium channel. But in filing the patent, some of the early patents were broad, and then they got more specific. And to our surprise, the patent office allowed some of the broad patents. So you've got to look very carefully to see if your patent is very narrow or is very broad. If it's broad, then it has more value. If it's narrow, then you've got to be wary of the fact that other people may be able to get workarounds.
Yeah, so it's pretty broad in the sense like mitochondrial dysfunction and neurodegenerative diseases. So it carries that aspect. Now, should I take this to the companies and do like a kind of presentation that that's my work? And if you're interested in...
Yeah, and you've got to be aware that there's a confidentiality issues. I mean, obviously patents are published so they can read the patent, so you can present anything that's in the patent. But if you have additional data, they need to sign a confidentiality agreement with you, and you've got to be careful of that. Of course, that's the way that you need to make a presentation. If I think large pharma try to be really clean. They don't want to get into big patent disputes, and they don't want any accusations they've stolen IP from anyone. But frankly, some of the smaller companies are maybe a little bit more risky because they right at the beginning and they'll say, oh, well, what a brilliant idea. We heard from him. Let's try it ourselves. And there's also enormous turnover of personnel. So new people come in and don't realize they got the idea from you coming in, and they think it came from their own work. And so in some respects, it's actually working with a large company tends to be cleaner because they will not cheat you. They'll have records of exactly what you've revealed to them, and if they have a license agreement, they'll hold up to it. But to be honest, there are some startups that are more fuzzy.
Thank you.
But they may be easier to sell a new idea to.
Okay.
Hi, I can go next. Thank you. That was very interesting. So my name is Celia McKee, and I'm a 4th-year grad student at Washington University in St. Louis. And I'm working on how circadian clock genes regulate functions in astrocytes in the context of Alzheimer's disease. And I guess I was kind of interested in when you were talking about bringing your experience to your mentorship. I was curious how your experience changes in terms of your mentorship style. If you have a trainee who says that they're interested in going into industry, what you see your role is in terms of helping them out. Do you think that that you should make them aware of what they should engage in the academic environment while they still have the chance? Or do you think it's more about pushing them to try sort of non-conventional things like going to groups like this or networking that they need to do since they're going on a non-conventional path?
Yeah, so I mean, it is a complex hybrid. In the end, my lab is an academic lab, although increasingly we do high throughput screens, and a big part of what we do is spend a lot of time developing assays and running screens. So we're like a biotech startup, except we publish everything. That's the output we have, and also we try and take risks. We try and develop new assays and new ways of. But and we can come to that in a moment. But if someone indicated to me they have an potential interest in going to industry, I'd say to them, while you're in the lab, you're an academic lab, and so you need to be exposed to my mentorship is going to be academic. However, I do encourage them then to attend meetings like this, to meet, to have informational interviews with people who have left academia and gone into industry, to see what it was like, to ask what is the path that worked for them, or what are the mistakes they made in terms of their career that in retrospect they would do differently. So it's more, I don't train them to work in industry or in startups, but I'm very happy to expose them to people who do. But you know, I hope that the work in the lab may be relevant. And for example, I'm part of a consortium funded by DARPA between Harvard Medical School, Boston Children's Hospital, MIT, and the Max Planck Institute. And we've been given 5 years to develop novel non-opioid analgesics. So it's an incredibly ambitious plan, because normally that takes 10 years. That's the standard time from, we had to start target agnostic. So we start just with knowing the kind of drug we want to develop, and the rest we have to discover. And to do so, we've really had to, it's been a massive endeavor. We've got to do massive omic analysis to identify differential expression of targets in the relevant cells. We've got to validate that. We've got to develop phenotypic screens as well as standard target-based screens. We are immersed into this AI-based medchem that we were talking about earlier, so that we can try and identify novel, using chemical diversity libraries, we can identify novel scaffolds with the desired profiles, and also develop completely new in vivo ways of looking at efficacy. So it's so we have a massive academic effort, but it actually parallels many of the activities that industry is doing. But as I said, the big difference is that our boss is DARPA, and we actually have to report to them every 6 weeks. And one of the rules when you get a DARPA grant is you're not allowed to use the same slide twice. You've got to keep on coming up with new stuff all the time. But we don't have venture capitalists or shareholders. So it's not people looking for money. It's a big, the Defense Advanced Research Program Agency looking for a success and pushing us hard to come up with new ways of tackling this problem. And the motivation for this is the opioid crisis. The best way to deal with it is to eliminate the need for opioids by developing non-opioid analgesics that are as efficacious as morphine or any of the opioids. So there's with this great alignment, we're aligned with the same interest. But it means that we've had to start cores in academic cores that are doing chemistry or that are running high throughput and high content screening, massive bioinformatics support. And so it's one of these cases where the world of our labs are changing. And the good old days when a single grad student or postdoc would have a single pipette and do one experiment at a time, that's history. Everyone now wants to do a minimum of 384 well format, so you spend your time trying to get assays that can run in this high throughput or high content, and always look into the imaging and where you can use robotics. We discover that every time you change the medium, you interfere with the cells. But if you do it robotically, the noise is much lower. So it becomes more complicated and more expensive, but you're sorting out these problems. And this is common to industry and common to academia. And it relates to, I think, a profound philosophical change in at least my approach to science. That, again, when I started, the overriding way of doing science was you had a hypothesis, you read the literature, you came up with an idea, you designed experiments to test it, and then you set about doing them. And frankly, that's the way you write your R01 grants to the NIH now. That's what your expectation is. Frankly, that to me that's history, because we now have the tools to do genome-wide screens, say CRISPR screens, or we have massive bioactive compound libraries. So you don't have to be restricted to the known data because it's incomplete. And so you want unbiased screens where the data will tell you the truth. It's not your ideas that are going to reveal, it's the data will tell you what's going on. What is driving Alzheimer's or Angelman syndrome? Yes, you may have ideas based on that will help you, but if it's truly unbiased, a true discovery approach where you can design assays that will reveal the truth, to me, that is the wonder of modern technology. And so I think that in doing so, we're using many of the tools that the industry is using, but using them in a different way. And so again, I think the academic filter is different from the industry filter, but in answer to your question, Celia, it's exposing people to new ways of thinking about things, new ways of coming up with solutions that they would hopefully be useful to them, even if applied in a different context.
Can I just ask a follow-up, since you mentioned R01s? Do you think that, I guess, academics interested in drug development need to write their R01s differently than sort of a hypothesis-driven project? Or do you think it's really just a matter of, grants will be reviewed in a certain way and they'll have that expectation, so it's kind of hard to come at it from an unbiased standpoint?
It really depends. I mean, NIMDS now has a big translational element, and which is run by people with industry experience, and there are RFPs, their requests for proposals are actually asked for details about screening, and so they encourage a more discovery approach with. But if you go to a regular study section where it's all filled with academics, there may be some who are willing to accept a drug discovery approach, and others who are locked into the standard hypothesis driven. And so, and to be honest, I think they're more of the latter. So I think you write your grant partly, and that's the art of grantsmanship, you write your grant partly for the reviewers rather than for the way you're going to do the work. And that's the big, I've only had this DARPA grant is the first grant I've had, and that was completely different. That was entirely this. As I said, we had to start target agnostic. So normally in R01, you say, I've discovered this new target, I'm going to prove that it's valuable, I'm going to have a screening pattern that's going to be related to the target. They insisted as part of the grant application, it was target agnostic. We had to, as part of this funding, we had to find the target and validate them and prove them, and there may be multiple targets. And we didn't know what the screens are going to be. We just had to be committed to developing certain kinds of screens, high throughput and high content, et cetera, et cetera. So that was a real eye-opener, where that was encouraging risk-taking and thinking big and out of the box. So it was very different from the standard NIH type of grant. And if those of you who are going to remain in academia, you will have to learn the skills of writing a grant, which is, as I say, in the end, it's science-based and it's got to be high quality, but it's very artificial. It's writing it for the reviewers rather than for the science.
No, thank you.
Anyone else have any questions?
Actually, I have a question. Hi, I'm Gautam Induri. I'm a fifth-year graduate student in the Pharmaceutical Science Department at the University of Connecticut Stores. My research is centered around a continuous manufacturing platform for improved quality of anti-cancer therapeutics. So we work very closely on translational and clinical aspects of research. As a graduate student, we aren't necessarily exposed to that area, and nor are we exposed to I guess, regulatory issues related to GMP manufacturing and or other regulations. So do you feel that graduate students may need another additional training or an additional maybe certificate beyond their just PhD studies in order to work on the late stage clinical aspects after graduation?
Yeah, it's I've got a graduate student now who plans to defend her thesis in May, and she wants to go into industry. And so she's struggling with exactly those issues. And the big debate for her is, should she do a regular postdoc? Should she do a postdoc in a large pharma company, because they do have fellowships? Or should she go into a startup? And this is where I introduced her to, I think, about 10 different either baby startups that have just started, some that are 3-4 years on and relatively successful with 10, 15, 20 people, and some that are very large, just to get a sense from them. And I think there's a strong feeling that once you're in there, you can learn those things. You don't need a course to learn what GMP manufacture is. That ideally, it's great to know about what it is and know where you want to be. But once you're in the industry setting, whether it's a biotech or pharma company, you will learn very quickly what it is that, the particular unique features that are essential for that particular function. And in the end, the feeling that the feedback she's got, that it's better to learn it on the job rather than to do a course, because they feel that they can teach you more that's going to be useful rather than just a theoretical exposure.
Thank you. And I just want to follow up with that. Would you recommend, or is your graduate student, has she received recommendations to maybe look at, say, a smaller company where she can work with multiple facets and learn different things, versus maybe a mid-size where they may have been established and she might have to be in a more defined role?
Yeah, and this is the same problem actually. When I have rotation students come into my lab exploring potential places to do their thesis research, I always say to them, there's no right or wrong way. Just make sure in your rotation that you have spent some time in a big lab, which may have the advantage that there are lots of things going on. There are lots of experienced postdocs. There may be lots of access to equipment. There may be multidisciplinary approaches in that lab. This is a small lab with new faculty, where the chances are the new faculty will be working right next to you, teaching you all the time. Whereas in a large lab, the chances are you will have 1 hour a week at most working with the PI. And so it's more remote, so you have to learn to be more independent. It's not right or wrong, they're just different styles. And it's the same, I think, for startups. And the advice she got was, it's varied. There's some people who love being right at the beginning in a new startup as it's tackling the problems, because there you learn how to what are the problems, what are the solutions, to have an active role in the actual construction of the company from the beginning, as opposed to some people who say, if you've got a more established company where they've sorted out all those problems, you can now get involved and learn the technical things and move forward. And it depends partly, if you're going to be someone who is going to be on the startup side, then you need to know what a startup is and learn how to manage it. If you're going to be someone who's going to be working as part of a large program in an established pharma or a large, Moderna-sized biotech, then that's less important. Doesn't mean that exposure to one can't help you later on, or vice versa. So I actually got, as you'd expect, mixed advice. Some people saying they think you should do one, and some the other. And in the end, she felt that she graduated from Harvard College and then spent a year in a small biotech, and then came to my lab, and she enjoyed the experience in the small biotech as it was growing. So she's biased towards that, but not completely. She's the other thing. It's quite different when I have graduate students who are looking for a postdoc job. I encourage them to reach out to labs about a year, because if you're going to have a postdoc job, they've got to make sure they've got a position available, or you apply for fellowships, and it takes a long time. Whereas when you're applying to a startup or a biotech, if they like you, they want you within 2 weeks. So it's a completely different time frame. So because she's only planning on defending in 3-4 months, all her interviews have been informational, and people have said, come back to us when you're interested, because when they see you, they want you. And it's something just to be aware of. The timeline of hiring is very different going into industry.
Thank you.
So I know we're getting close to time being up, and I want to be respectful of everyone's time here. So if you need to log off, feel free. Otherwise, we can, if there's anyone with a burning question to ask, we can take that with the last couple minutes here. Alrighty. Well, thank you so much, Dr. Woolf. I know there's thank you messages coming in the chat box as well. So we all express our appreciation for your time. And I think that's it for today.
Best of luck to everyone, and may you all find whatever career suits you best and be successful at it.
Fantastic. Thank you.
Thank you so much.
Time.
Thank you.